It's really my pleasure to be here. It feels, after more than 2 years of working in Costa Rica, it feels like my second home a little bit. So I'm really happy, and thank you, of course, also the managers of the exchange program for this kind invitation. And I hope to share with you some interesting information that was elaborated during the past 10 years. A lot of the work that I will show has been done here, but also some other work pieces that we did in Germany with PhD students there. So obviously this is a lot of work that has not been conducted at the University of Geisenheim, but rather at the University of Hohenheim before. And since Geisenheim is a really small town in Germany, I would like to show you where this town is located. It's kind of in the center of Europe, close to the urban region of Frankfurt, Wiesbaden, which is the capital of the state of Hesse. And on the outskirts, somewhere on the fields, Geisenheim is located. It's good for the students. There's not much distraction in Geisenheim. Oops. And so that's where we are. And the university was only has quite a long tradition, but not as a university, but rather as a research institute. Was founded in 1872 as a Royal Academy for Fruit and Wine Production, so solely focused on research. And in 2013 it converted into a university, so opened up for students, and since then it's growing and growing, gathering more and more students. Still small, less than 2,000 students are subscribed to the programs. I don't want to give too much detail because we have scientific things to talk about it. But our main focus is plant food production and processing. The main focus of this area of Germany is wine production. So mainly white wines from a grape called Riesling, very famous German grape variety, and that's where we are. And our plant food research focuses a lot on beverages, of course also on wine, but we're in the progress of opening up to other sources as well, and also to research beyond just beverages. So the department that I am the managing director of is composed of 4 professorships, which is one is myself, the Professorship of Analysis and Technology of Plant-Based Foods, and then there are 3 other professors with their fields, all under the roof of this Department of Beverage Research. And we are have a strong research team with 5 senior researchers, and 2 of them are with me today, which I'm very happy of. And Christoph shared the presentation yesterday. Janka will share one tomorrow. But we're also equipped with a pilot plant where we have an engineer and some staff working, and that's basically what we are in a very short nutshell. I'm going to start my actual content with really the basics. Probably you are familiar with some of the background. I just want to make sure we're all on the same page about what is color, because when we talk about pigments, which is the title of my talk, natural pigments, we should be clear about what is color. And it seems like an easy question. The answer is I try to give a very short introduction. As you know, that without sunlight there is no color. And the interesting feature about the sun is that it emits radiation of all wavelengths, right? From gamma radiation to all kinds of wavelengths. The interesting part is a wavelength range between 400 and 380 and 750 nanometers, which is the visible range region. So when this light comes down from the sun, all these wavelengths hit your brand new Ferrari car, and the car absorbs all wavelengths except for the long range wavelengths, in this case here, the red one, which hits your eye, and then this is what you see. So you see what has not disappeared. But this image, when I show it, usually you get the image that the radiation itself has a color. It doesn't have any color. The radiation is just a wavelength and nothing else. So where the color actually starts existing is in your brain. And how is the path to the brain? The light beam, the radiation of 700 nanometers enters your eye. You have some receptors in there, and these receptors, they can receive the radiation and convert a radiative signal into an electric impulse, which is transported to your brain, and your brain says, hello, this is red. And you have three kinds of receptors. You have one receptor there that is covering more or less the blue range, so short wavelength range. You have one receptor for the middle part of the spectrum, which is more or less a green receptor, sometimes also called. So, and you have a red receptor at the long wavelength maximum. And this is how we will talk about later on when we talk about pigments. We will need this concept of the wavelengths. And now, as we are all working somehow with foods, the question is, what does this impression in our brain, which has not existed before, right? Nature doesn't know color by itself as a physical principle. But in our brain, it does something when you look at food. And I want to share with you one experiment. This is a very famous psychological experiment related to color in food. It's Wheatley studies out of the 70s. And he organized a study which was masked as a dinner party. So he invited his subjects to a dinner party and said it would be a dinner in the dark. Maybe you know that concept. Some restaurants all over the world also offer that. You come there, it's dark, you cannot see the food. It was quite novel at that time. So the participants should describe how they feel when they're eating the food without seeing the color. Actually, Wheatley was a funny guy. So this wasn't actually the goal of the experiment. He was interested in something else. He wanted to turn on the light after participants had eaten for about 5 to 10 minutes their food, and when he turned on the light, people actually saw what they were eating. He had colored the food in extreme, abnormal colors. This is a reproduction of it, it's not the original image, so this is Photoshop. But the meat was colored blue, and the fries were colored green. And the reactions of the participants when they saw the plate was the actual goal of the study. And an extremely high proportion of them suddenly fell ill, with several of them heading straight to the bathroom. So the fundamental principle between color and foods is that color raises some expectations in us. And when these expectations are not met, you are disappointed. Or even extremely disappointed when it's totally not what you have expected. And this is important when you look at the evolution, for example. We use color to judge if a food is still okay, or if it's rotten. Or is the food still fresh? When you look at the peas, these terrible peas that they sell in cans, they have a terrible color. They don't look as nice, of course, like freshly boiled peas. Naturalness is a big topic worldwide, and you see, depending on the color, which one would you think is natural, colored with natural colors? You can immediately judge which is more likely to be colored. Or South American, very famous product, I think in Peru, Inca Cola. When you look at this, you immediately know it's not a natural color. And it's such a vibrant color. While in Germany, we like to drink truly organically farmed apple juice, and this color needs to be a little bit brownish, like nature is, not truly brilliant, but people expect that. Wine is also clearly, you have expectations when you drink white wine versus when you drink a red wine. You have different expectations, and these need to be met. And that's why coloring food is so important, because when we produce food for the supermarkets, they need to be stable for a long time, for the shelf life, up to 12 months. And natural pigments, normally they are highly labeled, so there's very few natural pigments that make it until these 12 months, at least as long as they're from the original food source. And that's why we need to color food, to restore the color that was lost during production, or to give it a new color. Think about the gummy bears or jellies, you give them a new color, and that's why it's important. And interestingly, in the earlier times, until the 19th century, coloration of food was done exclusively with natural sources, natural pigments, because there were no artificial dyes. So people had to use something that was, for example, sepia from the seas, or they used spinach to color things green, and they also used heavy metals like lead, salts, and stuff like that, which is also natural, but still, maybe not always healthy. And then in the 18th century, industrialization hit, especially in Europe and the US, and then the demand for for convenience food increased for the first time in history. And that's when also the demand for pigments increased. And there was not enough natural pigments. So the chemists, which were also emerging very strongly during this period, developed artificial dyes chemically. And the first dye is shown here, that's not shown here, is called Mobi, but which is from the type of aniline-based colorant, which is structured like this. And from then on, from this time, 1856, the artificial dyes entered the markets and were widespreadly used. And only about 1 or 2 decades, depending on where you look at on the planet, we see a trend backwards to natural pigments because people more and more reject artificial dyes. And why is that? Why are artificial dyes restored? There is a trend generally for more natural food, but for colorants there was a study called the Southampton study, which was really putting a bad mark on the natural colorants. Big human study that associated the consumption of artificial dyes with adverse effects on the activity and attention of children. So think about the gummy bears, and you have these effects on your children, you don't want to give that to your children. And in the European Union, they have to label artificial dye, they have to label products with artificial dyes with exactly this term. So these 6 colorants rapidly disappeared from the European markets because of that. And one of the pigments that was used for the red range is also noteworthy, I think. Was used for coloring all kinds of red foods. It was from an animal source, from Coccinilla lice, so an insect. It's a very stable colorant, one of the most stable natural pigments at all. But Starbucks had this problem that they colored their strawberry frappuccinos with cochineal dyes. And the problem was not, as you might think, that people didn't like the disgusting view on the bugs, maybe, or anything like that. The problem was that this frappuccino was labeled vegan, and the bug is an animal. So the vegans gathered and blamed them for selling vegan food, which was actually not vegan. This is how the scandal started in 2012. And then a lot of other people realized that the frappuccino was containing insects, which was then disgusting to other people, and then even other people in the science field realized that cochineal is produced with aluminum, a salt that has been associated with the emergence of Alzheimer's disease. So a lot of different communities then attacked the carmine, which really led to the ban of carmine in many, many European products at least. So in a nutshell, artificial dyes and dyes from animal origin are not really desired by the food industry anymore. At least in Europe, it's a very strong tendency. So that's why the whole field moves to plant pigments, not only now, since the past 2 decades. I mean, also Patricia has done a lot of research, not just since yesterday. And basically, you find all colors in nature. You find green chlorophylls, you even find blue pigments in fruits of juniper, for example. Or you find anthocyanins that have blue and red colors, you find betalains, you find carotenoids. So we can talk for days about colorants, all kinds of color ranges. We focused a lot on carotenoids because carotenoids, first you find them very frequently in nature, so there's a lot of good sources for that. You find over 750 different pigments. The most popular vegetables are colored by carotenoids. And interestingly, we know more than 750, but in our diet and in our bloodstream there's not so many. The diversity gets a little bit reduced, which makes the study more easy. So you see these 6 representatives here from beta-carotene, the most famous one, to maybe lycopene, the red pigment of tomatoes. And the good availability is not the whole story, of course. You have more than color. That's something very interesting, because instead of having adverse health effects with artificial pigments, you get health benefits with plant pigments. The most famous one is the conversion of beta-carotene to vitamin A. You know, vitamin A, first vitamin to be discovered, A letter, and associated with a lot of health beneficial functions. Don't want to talk too much about vitamin A. You know that it's important. And then there is a health benefit that is related to our eye health, because this is a very famous picture, if you look here, from Max Nodelli from New York University. He prepared a microscopic slice of the human retina, and there is clearly visible on the back part of your retina, about here in your eye, is a strongly yellow spot, which is there in all humans. Stronger or less stronger color, depending on the diet. So our human body puts these pigments there for some reason. And since we cannot synthesize them, we need them from the diet, to take them out from the diet. And it's thought that they prevent a lot of diseases because they are kind of a blue filter. So remember the light beams coming from the sun. Of course, they also hit your eye part of the cornea, and the lens absorb the UV radiation. Where if it's extremely excessively absorbed, you get cataract, you get first disease, because UV radiation has a very high energy. So most of the blue to red light beams, they pass through. And since the blue light and the purple light here has also very high energy, it is believed that these pigments help to diminish a little bit this radiation, to lower the damage that the blue light can do to your retinal cells, to the photoreceptors that we've seen in the first place. So there is good reason to think that we want plant pigments, because they seem like so much better than artificial pigments. The problem is that they are not as stable, and we need to understand how to stabilize them, how to produce the right colors with them. And that's the next part of my presentation, showing now the research, where we try to play with these pigments to produce different colors, play with these pigments to produce different bioavailabilities also for the health benefits, and how all that is related to the chemical structure. I want to first start with something that we did together with the university here, with Patricia and Victor. We're studying the actual deposition forms, because if you look at these pigments, they are not evenly distributed in a carrot. This is, I think, widely known that carrots contain crystals of these pigments. You see them here in the light micrograph, tightly packed molecule stacks that are arranged in this way. This is widely known since the 60s, and I think everyone knows that a carrot salad with a little bit of oil improves bioavailability. This is not something we were focusing on. I want to show you these two slides, because that's also where we kind of started to look at other foods then. And when you, there's one first relationship connected to this crystalline point. When you look at a lot of different fruits, like pink grapefruit, papayas, also the red one, then you see crystals like this always, or almost always, when you find these two pigments, beta-carotene and lycopene. They are highly prone to forming such crystals. And it is believed that the crystalline form fosters the crystal formation, the molecular shape fosters the crystal formation, because these molecules, if you take molecular models, they fit nicely together, especially lycopene. This is a lycopene model. It's like a stick, and you can place them very closely together, about 2.5 angstrom of a distance, which is virtually nothing, so it fits very nicely. The beta-carotene is already less tightly packed. You get 5 angstrom, which will be later an important measure of the distance. So this first observation is that when you have this kind of pigment, you see these crystals very abundantly. So that was a relationship first in the first place we saw. Studies that were done here were now, when we looked at the peach palm fruit, we were thinking there must be something where the oil in the carrot salad needs to be prepared by nature already. The hibayi, peach palm, was one of these fruits that Patricia discovered, or Victor with a student that was working here. And this fruit, which contains a lot of fat by nature, apparently there were no crystals. There were like these little droplets here, chromoplasts are they called. And internally they again have little droplets. You see a light transmission electron micrograph here. This is a chromoplast, and you see these little lipid droplets in here. And that's also where you see this strong color. So nature has prepared a form where these pigments are already dissolved. We also found that in tangerine tomato, which has, interestingly, has no lipids at all. This is common tomato, less than 1% fat. But still it contained these little lipid droplets. Was a myth a little bit. And the key to understanding this is, when you think about the molecular shape of lycopene, the major pigment of tomatoes, in this particular tomato, the lycopene was bent a couple of times. It was deformed. So about 4 C configurations, which leads to, in comparison to the normal lycopene of red tomatoes, which leads to a bent molecular structure, which then results in the fact that you can't put them so nicely together. So crystallization is really inhibited. And this is why we believe we find no crystals. It's the same pigment, and but a different aggregation form. And this is when I thought this is such a strong influence also on color. It's the same, basically the same pigment, but truly different colors. Concentrations are similar, so it's not solely a concentration effect. It's a little bit higher in the red tomato. And this is when it's becoming interesting for food industry, because this can be used to play around with. And my former job at DSM showed that it's really a key point to play around with the physical state. If you have a beta-carotene that is truly dissolved in oil and you measure its absorption spectrum, right, from the blue, I simplified a little bit, the blue, the green, and the red regions, you see that the absorption of beta-carotene is mainly taking out the blue portion of the light, and the red and the green portions are reflected, which leads to the impression that it's very yellow. It's not a natural feeling that green and red gives yellow, but try it with laser pointers. You can see that if you have a green and a red one, it gives yellow. So this is what you see here. You see yellow colors. On the light micrograph, you see nothing because the lipid droplets are very small. And on the other hand, if you now take the same pigment at the same concentration of 10% and you crystallize it, and you again then measure the absorption spectrum, what you notice is that although it's exactly the same pigment, you get a different absorption spectrum. All of a sudden we have an additional absorption band here, right in the middle of the green region, taking out now the blue and the green stuff. And only red light is reflected. A little bit of the green also gets through. That's why it's not a brilliant red, but it's more orange, reddish tone. So with the same pigment, at the same concentrations, you can produce extremely different colors. It's highly interesting for food industry. And the question is, now playing with color and the aggregation form, what's the impact on bioavailability? We took these, that's maybe something that I showed earlier here in 2015 or so at my last talk, which was a human study that was conducted with the red and orange tomatoes at the Ohio State University, where I was at that time. They did the study. They produced tomato sauce or soup from both tomatoes and then fed it in a crossover design to 12 participants and analyzed how much of the carotenoids come to your bloodstream. So what you see is the concentration of the carotenoids over time, and when you take the red tomato soup, virtually you get nothing of the lycopene. It's very, very low. So the crystals, they are of course mashed up, they are smacked down to smaller sizes, but still they are crystals, and our body cannot absorb these. If you would add lipids, okay, it would be different, right? But interestingly, when the tangerine tomato with the naturally lipid dissolved lycopene was fed, we see an absorption curve like this. It's like you inject it straight to the bloodstream. And what you measure normally is the area under the curve, and then compare the areas, and you notice that this is 8.5 times, 850% more lycopene is coming to your bloodstream when you consume this tomato soup. So Campbell has produced a tomato soup on the US market. And they cannot advertise this, but they also think that the color is nice. And apparently people are buying it. So lipid dissolved carotenoids have really a strong impact so on bioavailability, but also on color. And the question is now, the bioavailability part was known already, but what was not known is that is there something in between, because it's two extremes, right? Lipid dissolved and solid crystalline, and this was actually something that we did together also. Was we discovered something that is in between on papaya fruits that we collected in Costa Rica. We collected them and we analyzed by light microscopy their chromoplasts, and when we looked at them under the electron micrograph, I think which was produced here, this micrograph in the ceramic facilities, we didn't see any crystals, we saw some lipid droplets, but interestingly, we saw some other elements, like these tube-shaped forms, which were later on then in the group, also found in other berries. The nicest chromoplast photo I've taken is from goji berries, very popular in Germany for muesli and breakfast cereals, showing lots of these tubes, and that's the place where the carotenoids are located in a liquid crystalline form. So there is something that is in between lipid and solid crystalline. It's a liquid crystalline form, right? Something in between. And it has not been studied much. Some biologists in the 60s and 70s have described it in peppers and speculated a little bit about it, but then it disappeared again because no one was interested in natural pigments. Remember, at that time, people were dyeing their foods with artificial colors. So it has changed now, and what we try to understand, and this is most of the part of my habilitation thesis, is to understand why is this liquid crystalline state, when is it coming? Because we've seen earlier that solid crystals are related with beta-carotene and lycopene very often. Lipids dissolved is also imaginable. If you have lipids, okay, you can have a lipid dissolved from them. What about the liquid crystals? And so we studied a lot of fruits and vegetables and also gathered information from literature. Some of the information is from the 60s and 70s, for paprika or peppers, for example. But if you look at all these fruits where we find liquid crystalline aggregates, it's not a curiosity in papaya, right? You also find it in mango, you find it in pepper, physalis, you might know. I think Tanya has found them in mamey, zapote here. You find them in a lot of different fruits. And the thing they have in common is that all of these contain carotenoid esters, so compounds. It's the carotenoid, and then you have fatty acid, fatty acyl moiety at one or both sides of the molecule. This was something that was there in common, and which was the first interesting observation. And the thesis of Judith Hempel, which some of them might know her, was exactly focusing on reproducing carotenoid liquid crystals in vitro with carotenoid esters. So what we did then was first, and it was a lot of work, isolate and purify carotenoids with different structures, all kinds of different structures, partially synthesize those that we could not isolate from nature, and we had in the end zeaxanthin monoesters, a variety of different molecules, because we wanted to study every feature of the molecule, look at the influence of the OH group. What if we take out the OH group? What if we esterify one of the OH groups? What if we esterify both of the OH groups? And once we had the collection, we took all these purified pigments and conducted a controlled aggregation experiment in vitro by slowly evaporating a solvent. That was one form. So you concentrate the pigment more and more, and at reaching saturation state, it starts crystallizing. The other form how you can do that, which we also did, was to add water to a water-miscible solvent. So you increase the polarity of the solvent mixture, and at a certain polarity you also reach saturation and you get aggregation. You get a so-called lyotropic aggregation. And then we studied these aggregates that we had formed to see if we can find similarities to those found in nature by spectroscopic techniques, UV vis and circular dicrystal spectroscopy. I want to show you some of the results when we use the main pigment of goji berries, zeaxanthin dipalmitate, which is esterified on both sides. We study the absorption spectrum in solution and in an aggregated form. So what we also see is that we get such a J-band, as we've seen before with aggregated beta-carotene. And from these spectra, I can't show these details, but from the spectra itself, you can calculate the intermolecular distance. Because the closer you push these, the more shifted gets the absorption band. So you can actually really calculate the angstrom distance. And you can also conduct molecular models by computational modeling and see if that matches. And apparently, it works quite well. We have 1 to 2 angstroms of a difference, 12 angstrom difference. And that was interesting, under light microscopes, we saw tubes. So they were moving sometimes, and we thought, that's already pretty close. They were much larger than those we found in papaya or goji berries, but they were there. They were tubes. We found the same thing when we had only one of the OH groups as terrified. We also produced J aggregate. We also produced these things here. When we take out the whole group here with cryptoxanthin, interestingly, we also get the same spectra, or similar spectra, but all of a sudden it shifted to more solid crystals, rhombic, large crystals, no more tubes. And the distance calculated and computed was around 5 angstrom. So apparently, the molecules got closer together with the cryptoxanthin. And we thought that makes sense, because if you have a long acyl moiety of fatty acid, which is a big chunk of matter, just in between the aggregates, molecules can't go so close together, and when you take that out. And then interestingly, when you put back the OH group here, the spectrum completely changed, because the spectrum then didn't show any J band anymore. It shifted to the left side, so color was disappearing, not completely, but strongly weakening the color. These aggregates are also known, not for carotenoids, but they are called H aggregates, for hypsochromic shifted aggregates. And what happened here is we found also crystals, but the intermolecular distance, when you look at it, was very, very close. And the idea is, or was the hypothesis, that we now have hydrogen bonding between the different molecules. So when you have two of these molecules, the hydrogen, the OH groups might form hydrogen bonding and pull the molecules together. And this only works if you shift them a little bit, if you distort, if you have an angle of distortion. And then you can push them even more together. This is shown by molecular modeling here. And then you see here a very thin green line, hydrogen bonding between these molecules, which brings us below 5 microns and changes these colors extremely. If this is right, then the molecules should form a helical structure, because if you think this consequently through and stack one of the other, you should get a helix. And helical structures can be studied by circular dichroism. DNA, for example, shows strong signals in circular dichroism. And these aggregates, interestingly, also showed strong signals in circular dichroism. So we were pretty confident that our theory has some support. And so this is an image of that. We did that with a lot of different compounds. There is an article about that because it's quite heavy to tell that in a short amount of time. Interestingly, it was completely independent of the chain length. So if you use the C2 chain or C18 chain length, it didn't matter. You just need a small spacer, and immediately you get these liquid crystalline tubes. You should not. There was no difference in all these differently synthesized ester. If you have one or two esters, that doesn't really matter. And then we tried to use this knowledge now. We thought using an ester, whatever it might be, is the key to the liquid crystals. We want to do that technologically because we're food technologists and we want to make something out of this. So we isolated from goji berries a big chunk of zeaxanthin dipalmitate in a big vessel, so a kilo of pigment. And then we used a certain process, I don't want to go into the detail, which is producing lyotropic aggregates, so water-miscible solvent is used. And basically you have a water-miscible solvent and you mix it with a water phase, also composed by certain stabilizers and antioxidants. So there's kind of a recipe behind all this. And then we produce the aggregates to reproduce these liquid crystalline form. And it worked. What was the difference was that we could produce with zeaxanthin dipalmitate, J-type aggregate, something like this, which had a strong color, very intense, and with pure zeaxanthin we were producing the H-type aggregate, which was a very light color. So it's the same concentration, the same core carotenoid zeaxanthin, with extremely different colors. And then we put that into a human study, same design as earlier, so we had crossover design, 16 subjects. We gave them the preparation, which was a drink, and fed them to 16 subjects, seeing that the H aggregate, remember the one with the very tightly packed carotenoid aggregates, seeing such an absorption, and then the J aggregate, which had a higher availability, a higher area under the curve. Honestly, when we looked at the curves, we were a bit disappointed because we thought the impact would be bigger. Remember, 850%. From lipid dissolved to solid crystalline. Here, from these two aggregate types, we only find about 23% difference. so not too much. Now, I want to come back to this because this is an artificial product, right, that we produced in the laboratory. How about that in nature? Can we transfer this knowledge? And this is a very early study that we did, actually before the technological studies. What about the liquid crystals from papaya that I showed you versus naturally solid crystalline aggregates? And here the differences were much bigger. So papaya presents a very, very good source of carotenoids, having them in a liquid crystalline form. And carrot and tomato are very poor sources, which was already known. This was a nice finding for applied science because you can eat papaya, which presents carotenoids in a very good form. But apparently there's more factors than just the aggregate effect. So you have a softer tissue than you have in carrots, and you have a different matrix. So the bad, and I think the disadvantage of this study was that we had completely different matrices, although looking at the same pigment, beta-carotene. And with the technological study, we had the identical matrix, but we had differently aggregated pigments and different pigments. So you can't have the perfect study, but if you take all these two together, I think we have a reason to believe that there is a kind of a ladder for bioavailability. You have the solid crystals, you have the liquid crystals, and then you have the lipid dissolved. What if you like carrots? You still want to have the best out of them? I talked about that. You can also help on that. I just want to show a study where, to make it more real, that was conducted earlier, also at Ohio State University. And I selected it because aguacate is a very popular fruit here. And if you combine this with the carrots and then do a human study as we did before, you can see interesting effects. If you eat only carrots, the absorption curve also runs like a flat line. You can virtually get nothing, almost nothing out of it, barely above the detection limits. And when you add the avocado, which was in this case fed as guacamole, you see a substantial boost of the bioavailability. Same happens to the vitamin A production, so you not only get more beta-carotene from it, actually your body can make use of it and produces the vitamin A, so it's actually true benefits. And part of the study that then we did in Hohenheim at that time, we were focusing on other factors because we were really curious to understand all the matrix effects, and we were looking at other factors. One study that I want to share with you is also related to a product that's produced in Costa Rica. We thought, what happens to the carotenoid bioavailability if we take an orange and we simply dejuice it? So the effect factor would be, the matrix factor here would be the dietary fiber, the cell walls. How does that impact bioavailability? So we produced same design as always. We fed people with oranges, crossover design, randomized order, and then we fed them with orange juice and looked at the carotenoids in the bloodstream. And interestingly, fiber is a factor that counteracts the bioavailability consistently for all carotenoids that we looked at. Here, for example, beta-cryptoxanthin, the bioavailability from the fresh fruit was lower than from the orange juice. This was consistent for all carotenoids, maybe not so surprising. Zeaxanthin, lutein also. Orange juice was a very, very good source. And when we published these results, I did not expect that it was hitting such a media response. But media was telling now the story that orange juice is more healthy than fresh oranges, which was a shocker for many people because the fresh fruit is the golden standard. And this was extremely important at that time because the media hype in 2014-15 was exactly the opposite. I don't know how that was perceived in Costa Rica, but I extracted some articles from that time when we just had published a study about orange juice. The media was picking hard on juices. So there was this one article which I also found had much attention. Orange juice is just as bad for you as Coca-Cola because of the sugar content, right? Orange juice has about 10% sugar, Coca-Cola too. So the juice was in the dark corner and everyone was hitting on it. And we were just publishing that orange juice is even more healthy than fruit. So we got into a big struggle at that time. And the media was, it was not only the media, it was that in the UK they banned fruit juices from kindergartens and primary schools. So there were actually legal actions on that and to ban it, and they want, still it's ongoing, this act. And we thought that's not what we're seeing because orange juice is obviously so much different than Coca-Cola. We kept on with our study. we used the samples again and we said, if carotenoids are taken up better, then what about the polyphenols? So we look at the hesperidin and just extracted the same blood samples again and see what do we see for the polyphenols, hesperidin, and also some others. It's a bit more complicated because you need to look at the metabolites too. They're not absorbed in an intact piece, so they're fragmented into some major metabolites and some minor. Some of them are not even known. And this was important also at that time because the polyphenols from orange juice were related with some health benefits, surprisingly. So exactly counteracting the media perception. So they were related with alleviating the metabolic syndrome or weight gain, fat gain. So hesperidin was kind of a holy substance. And that's why we focused also on hesperidin. When we looked at the results, which are shown here, you see in black the fruit response, and in gray the response from the juice. You get a little bit more from the fruit, which was not significant statistically. Here it seems to be partially significant depending on the test you do. But the key difference was that you see, let's say statistically the same response or a very similar response, but the content in the fresh fruit was much higher, 2.4-fold higher. So you eat a lot more of the substance from fresh fruit, but you get the same as if you drink a glass of orange juice. Again, this was totally saying that orange juice is not a bad food. At the same time, the authorities went on, and France and Great Britain, they banned not only fruit juice from primary schools, but now they have a tax on fruit juice, and not on fruit juice, sorry, and soft drinks, depending on the sugar content. So all that has more than 8 grams sugar, you pay an extra tax of 28 cents per liter, which is quite a substantial per 100 milliliters. So that's a lot. And in this range, you get 21. So the sugar was getting more and more into the focus. And we were still exactly on the opposite side, and that's why I now leave a little bit the color thing for the next course. I hope we have some more 5 minutes. Because we wanted to better understand now, coming from carotenoids, is orange juice really the same in terms of sugar also? Because we see that the hesperidin is there, the beta-carotene is there, and there's a lot of other substances there. I have a list here. If you compare orange juice and Coke or any other cola drink, sorry, no offense about Coke, same for Pepsi and others. But you see that you get a lot of potassium with it, you get vitamin C, you get the B vitamins, you get folic acid, you get vitamin E, and in the soft drink you get nothing of this. So we hypothesized that the metabolic response to orange juice could also be different than to cola drinks in general, not just on the carotenoids. And we did a study with nutritionists from Hohenheim, where we fed people 3 times a day for 14 days with orange juice, and they drank about half a liter of this drink at each instance. So you get quite a lot of orange juice, 1.5 liters. And the same then with Coke, or cola, sorry about that, for 2 weeks. Randomized crossover design, so everything like this in between the meals. And then we looked at the metabolic response, or the nutritionists looked at what happened with the participants' blood profile. And I want to move forward quickly. The first curve that I want to show you is here, up here. So you see this is a day-long glycemia sensor. So participants had a sensor on their back, and that is measuring the interstitial glucose concentration for the whole period. And when you look at the collective data of 26 people, you see nothing. So it looks really similar. But when you look at the single participants and compare their orange juice intervention and their cola intervention, you see an extreme difference in all of the participants. When they drink the cola beverage, they have really extreme blood sugar spikes. And with orange juice, the response was very moderate. The spikes that you see were much less pronounced. This can be summarized in an indicator. The response, the spikes were significantly more pronounced in all 26 participants here. And also the area under the curve, this is what you see here, is much higher. So apparently you not only get the sugar more quickly and more like an injection, but you also get more of it from the orange juice. And then maybe the last slide on this off topic from away from the pigments. We also saw enormous difference on uric acid. So a very important blood marker when it comes to the development of gout, which is a very severe disease worldwide, related with obesity many times and also with high sugar consumption. And what you see here is the uric acid level of the participants before the cola intervention and after the cola intervention. It's the same, surprisingly actually, because we thought if you drink 1.5 liters of cola every day for 2 weeks, there must be worsening. It wasn't. So the young students could easily cope with that. But interestingly, when they did that with orange juice, the uric acid levels, which we thought could only go up, even went down. And I remember that day with Anja Bosi-Westphal. She could not believe these results and she had to triple check because she was hypothesizing that orange juice is as bad as cola. And now she saw that the level goes down. It looks like a tiny difference, but this is because you see an average distribution here. So you see participants that go from here to here, you see them going like this, and all these that are in this sector of the evaluation show a significant response, a significant lowering effect to the uric acid. And we're trying to also follow up on this and understand why is this response so different. There were other factors too in the metabolic response. I think I'm over time anyways already. But the metabolic response is dramatically different. And that's why we want to investigate deeper now also in Geisenheim. What is it that makes orange juice so special? Because we could learn from orange juice and then transfer this knowledge to soft drinks to make them better. That's something that we would like to better understand. There are some hypotheses that it is related with the hesperidin or with the vitamin C. It's not clear at all, and we would like to look at that. So, just a brief summary of this now for the last 50 minutes. What might be take-home messages would be that plant foods are actually containing a lot of healthy compounds, especially the carotenoids, our true beloved. But it's also that these compounds are not all the same. They actually show the highest diversity among all the natural pigments, not only in terms of molecular structure, but also in terms of deposition form, colors that you can produce. Even with one single pigment, you can produce a range of yellow to red. Bioavailability is dramatically different. Lipids enhance, dietary fiber slows down. So you can actually, as a technologist, play with the carotenoids quite a lot to steer color, bioavailability, and so on. And the last side note is that orange juice might not be as bad as cola for you. I would like to know later on from you how this is perceived in Costa Rica, but in Europe the situation is dramatic. They're hitting very much on the orange juice still, and on juices in general, and I think this needs a more diverse, more deeper look at least, because it's clearly not the same. So thank you very much for your attention, and I hope we have some discussion and open for questions. Thank you very much for having me again. And
results for advertisement. What was the reason for that, or when comparing the two tomato types, tangerine and the normal tomato?
It's they could say it's a more bioavailable form, but they cannot say anything that's health related. And this is what the consumer would expect, because what does that mean, more bioavailable? Is that good or bad for me? Actually, I'm not sure what were the reasons. And maybe they changed meanwhile. I have not checked on the internet yesterday, so it might be that it has changed, or maybe the product even disappeared. I think it's still on the market, but there was the regulations on health claims are very strict also in the US. In Europe, it's even more difficult. Lycopene was associated with some health benefits related to prostate cancer, but lately, the last 3, 4, 5 years, evidence has fallen apart. There were studies that showed that there's no effect. So I think the lycopene community has been struck hard by a bit of a disappointment.
What about the antioxidant activity of this tetra lycopene compared to the?
It's very high. It's like as lycopene is very high. But studies that are looking at that, they very often struggle with the poor solubility of the ultrans or Ali lycopene. And sometimes it's not clear if the studied lycopene was crystalline. Crystals have no antioxidant activity because they're a rock, they're solid. And the tetracis is much more accessible, it moves faster, it doesn't aggregate. Even in solution you have aggregates, right? The tetracis doesn't do that, so it's much more mobile. So, but it's very, very good antioxidant too. Yeah. Yeah, please.
Which carotenoids are used now in the industry for as pigments?
So there are two major fields where carotenoids are used, and one is human nutrition, obviously, that was what we were talking about, but the bigger market is animal nutrition. So for salmon and shrimp farming, you need carotenoids to produce the red color of salmon and the red color of shrimp. And these are normally produced synthetically and then fed to the farms, to the aquaculture farms. That's bigger, a super big market. And the second biggest market is then beta carotene for foods. It's used everywhere. I think the butter that we had at the hotel is colored with beta carotene. You'd be surprised if something else, because it's cheap, it's readily available, it's healthy. So industry really likes to use beta carotene.
And is natural or synthetic?
Both are available. We get synthetic one, which is very cheap, and you get natural ones extracted, for example, from palm oil residues, from carrots, or, and this is the major proportion, biotechnologically produced version of beta carotene, produced with a fungus, Blakeslea tree spora, which is an interesting fungus that upon sexual mating produces beta carotene. So it gets red, it gets shamed, and it turns red. It produces extremely high concentrations of beta carotene, and that's technologically used and produced. And the other carotenoids are canthaxanthin, which is used for eggs. So also animal nutrition, right? You give that to chicken and the yolk gets colored red or orange, depending on the concentration. We have lutein, zeaxanthin in the markets, very much in the dietary supplement industry. It's marketed for eye health very strongly in the US and Europe also. It's heavily advertised, and I think these are the major ones. There's still a lack in the bright red region, so yet there's no carotenoid on the market that can mimic the color of carmine, of cochineal, which need something that needs to be discovered, I think, because there is no alternative still.
What about natto? Cement.
Yeah, it's also
being used.
It's on the markets, yeah. There is a company in Denmark who's marketing it. It's mainly used for cheese. Cheddar cheese, for example, is dyed with natto. This English cheese, they like it's a strong color cheese. Yeah.
Is naturally extracted or synthetic?
It's natural extract. I think these were the major ones.
And do you know if in the market there's capsules, carotenoid capsules, as supplements for?
Yes,
It's
yes.
not the final product with vitamin A, but
Pure carotenoids?
pure carotenoid.
No. It always needs to be diluted a little bit.
Yeah.
Because you need milligrams
Yeah,
only.
some oil, but I mean it's carotenoids has main components.
Well, yeah. You get that with oils. And it newly entered in infant formulas. Infant for if you do not breastfeed, then you can buy infant formulas. And earlier, the carotenoids were ignored because actually that's was surprising to me, because since I have babies, I know that the first breast milk that's coming out is deeply yellowish colored because of lutein and beta carotene. So there's strong evidence that suggests that babies during the early phase, they need these carotenoids, because when there was a study which is very recognized on preterm death infants, so infants that died, and they looked at their brains, and the major difference was that the carotenoids were depleted. If this was a consequence or a cause, it's not clear, but simply the chance that it could be a cause led all infant formula manufacturers to buy lutein and put it into the formulas, because it doesn't do any harm. It's in the natural diet. That's a big market for it.
And is any difference in preferences between synthetic and natural carotenoids?
Depends
for
on the market. In
different
Europe,
uses?
in Europe you have natural, very high demand to natural pigments. And in the US not so much, I think. In the Americas I'm not sure, but it's not that strong. I think Europe is really pushing for natural pigments. It gets a drawback now because I read a study comparing the CO2 impact, the carbon footprint of natural beta-carotene versus synthetic beta-carotene. And the one of synthetic beta-carotene is an optimized chemical synthesis since the 40s, 50s, optimized to the bone, and very efficient. While the natural beta-carotene from Blakesley at Riesborough, you need to grow the fungus. You have huge bioreactors that need to be heated, warmed up. Then you need the sexual hormones that needs to be produced. And you put that into it. You have to wait again, heat again, harvest the fungus, extract the fungus, purify the pigment, formulate the pigment. It's an extremely long process, so the impact is high. I'm curious to see what happens now, because now they have to weigh out these two trends. Natural, do we want natural, which is really bad for the climate, or do we accept some chemical synthesis? Very interesting controversy. Not only in the field of carotenoids, this is true for many other sectors of the industry too. Yeah.
I think the viability of the carotenoids depends on the ripeness of fruit. For example, if it's more ripe, then the viability is higher, but this is not related with ripeness.
I'm not aware of studies that tried that, but I would, from the knowledge that we have, I would speculate that the ripe fruit should be more bioavailable. Because now what you get is, during fruit ripening, tissue softens, so the release is not an unripe papaya, like a piece of wood. And then also pectin gets degraded, so the dietary fiber gets cut down. And an effect that you usually see is that simply the concentration is higher when fruit is fully ripe. With papaya, this was, I think we did that together, right? It's showing a nice developmental course.